Hello, my dear students. How are you all? I think so. You all are keeping very fine, healthy, and like I would say, in a jubilant mind itself. Okay. So myself, Shamitra sir, again back with like IIOCS program, and today we are going to focus over that of what? Today we are going to focus about that of like the part that is the thing that is the another kind of the bacterium, and that bacterium we are representing it as out of what? We are representing it as out of the Escherichia. Like I would say, in a jubilant mind itself. Okay? Just 1 minute. So myself, Shamitra sir, again back with like IIOCS program, and today we are going to focus over that of what? Today we are going to focus about that of like the part that is the thing that is another kind of the bacterium. And yes, there were some issues. So we are again over here itself in that case. Okay. So as we were discussing, we are going to like focus over that of what? Today we are going to focus over that of like the Escherichia, the Clostridium, and these two kind of that are the genera itself. Okay. And we are going to see the different kind of the morphology, the structure, along with that of what? The toxins which are being released by that of Escherichia Clostridium. So, we are again over here itself in that case. Just wait, beta. Just there is some technical issues. Just one. Just wait. Okay. Yes. So we can again start. There were some technical issues from my end only, like the part itself. So we will discuss the part. So today we are focusing towards out of the like the toxins or the morphological sections, okay, which are been released from the of the Escherichia itself along with the of the Clostridia. Okay. Now, if I'm saying about the of the Escherichia part, okay, Escherichia, we are knowing that it's a kind of harmless kind of the bacterium. It is generally a human friendly kind of the bacterium itself, okay, which is remaining present within that of what? Which is remaining present within that of our gut, which is present like in an inherent condition within that of our body system itself. But if I'm discussing about the Clostridium, Clostridium is an inborn pathogen, a harmful pathogen, I would say. Okay, which causes deadly kind of the disease, and even which releases a specialized neurotoxins also, which releases a specialized neurotoxins also over there. Okay, and which cause like around paralysis and many of the other kind of the features also in that case. Okay, so we will start first of all with that of this harmless group, that is the Escherichia. Okay, now this name Escherichia, Why this name Escherichia is present over here? Escherichia name, it is because of that of what? Because of that of a scientist who had first of all discovered that of this group of bacteria. Okay, and that name of the scientist was Escheri, based on which we are representing it as that of the Escherichia. And the bacterium which is living within that of our gut, within that of our body system itself, we are representing it as coli, Escherichia coli. Why I am saying it as a coli? Because colony forming, we are representing it as that of a colony forming bacteria. So hence Escherichia coli itself. So do remember one thing, students, that this bacterium, it is actually a purely typical one. I would say that a harmless kind of the microbes which is present within that of our body system itself. And once it is present within that of our body system, it actually what? It actually like it is acting as a, it is showing a commensalism kind of relation within that of our body itself. Now, like whenever it is getting any kind of that of the favorable condition for that of its multiplication, automatically what will be happening, the Escherichia coli, it will be getting like stimulated to show faster rate of multiplication. It gets multiplied, and automatically within that of our body, different kind of abnormalities it can get occurred itself. Okay, so let's see some of the features of the Escherichia itself. Okay, it is the universal inhabitant of the human gut. It is a universal inhabitant. Now, what does it mean? That stands for what? I would say that in all the human being, in their gut, in the alimentary canal, the Escherichia coli is remaining present. And even not only in that of the Homo sapiens species itself, it is also found in that of other warm-blooded animals like body systems itself. And that is a harmless kind of the other group. So for that reason, it is remaining present. Moreover, it is a facultative and aerobic kind of the bacterium. Okay, we had already discussed earlier also that what is facultative anaerobe, what is facultative aerobe, what is like obligate anaerobe, what is obligate aerobe, like that. And it is also known as that of a harmless commensal. I had said you just few seconds back only, we were discussing that how it's a harmless commensal, as because this is what this is. I would say that it is remaining in a complete commensalic reaction within that of what, within that of our body system itself in that case. So we are representing it as that of the harmless commensal. Okay, now it is not causing that of the disease, or it is not causing any kind of abnormality within that of our body unless until it is stimulated to multiply in a huge abnormal number itself. Now we are representing it as an opportunistic pathogen. Why I'm saying opportunistic pathogen? As the same reason. Why opportunistic? Because whenever like they're getting any kind of favorable environmental condition, automatically E. coli, it's like its number will get raised. And once its number is getting raised, it will be showing a huge number of the infections itself in that case. So we are representing it as of the opportunistic kind of the infections itself. For example, we can see the gram-negative sepsis, the urinary tract infections, and all these things that is remaining present. Okay. It's also a fermentative kind of the bacterium. Fermentative means it shows fermentation. It shows actually the fermentation itself. So for that reason, we are saying it as the fermentative kind of the bacterium. Now, this one is that of a rod or that of a bacillus one. Okay, and it is a gram-negative group of the bacterium which is remaining present. Now, it is belonging to that of the group which we are representing it as of the Enterobacteriaceae family. Now, what is entero? Entero word, wherever we are getting, that stands for gut. That stands for that of the alimentary canal itself. So belonging to that of the Enterobacteriaceae family, that stands for that of what? It will be the kind of that of the group which is actually found in that of what? Found in that of like which is inhabiting within that of the gut itself. Okay. For that reason, this group of the bacterium, we are representing it as that of the Enterobacteriaceae family. These are typical mesophiles. Okay, they are like the terrestrial kind of the bacterium. Okay, which are the terrestrial kind of the bacterium which are remaining found, and they are perpetuating that of within a temperature range of that of 7 to 10 degrees Celsius itself, and the optimum temperature it is of 37. So that means what? Our body temperature. They can very well grow within that of our body temperature itself. Okay. So now next we'll be seeing that there are, you have to remember that there are certain categories of that of these E. coli. Okay, based on that of the infection that it is causing, the diarrheogenic E. coli. Now, what is the meaning of diarrheogenic? See, these are all the enterobacterial group. Now, whenever they are the enterobacterial group, they are always associated with that of what? They are always associated with that of causing diarrhea. Okay, they are always associated with that of causing diarrhea itself. Okay, so hence these E. coli, we are representing it as that of the diarrheogenic E. coli itself. We are representing it as that of the diarrheogenic E. coli. So if I am saying about the diarrheogenic E. coli, we are having four major categories of the diarrheogenic E. coli. The first category which we are representing it as, named as that of the enterotoxigenic, which is responsible to release the toxins. Okay, the enterotoxigenic kind of the E. coli itself, and this E. coli, it is actually showing that of the mild diarrhea kind of the condition, along with that of the severe cholera kind of the syndrome itself. Along with that, it can also show watery stools or without blood or mucus itself. Okay, and stomach pains, yeah, vomiting, these can get occurred. And the second group, we are representing it as entero-invasive E. coli. Now, what is the meaning of this invasive? Invasive means what? The attacks are alimentary canal. The attacks are gut. When they are attacking our gut, they are causing certain, because they are the rod one, so they are causing bacillary dysentery kind of the symptoms itself. They are causing bacillary dysentery kind of the symptoms itself, like fever, like abdominal pains, like watery diarrhea, and all these things itself they can cause over here. These things we are representing them as that of the entero-invasive E. coli. Okay, so now you have to remember one thing, students, that this entero-invasive E. coli, which we are representing it as EIEC, this entero-invasive E. coli, it is actually what? It is actually, I would say that the one which is showing very much similar kind of the symptoms with that of what? Showing very much similar kind of the symptoms along with that of like the Shigella bacterium itself. We had already discussed about that of the Shigella bacterium earlier also. Okay, so I would say that it is showing very much similar kind of the characteristics along with that of the Shigella bacterium itself in that case. Okay, so if I'm saying about the Shigella bacterium, it was only the difference between these two. The Shigella bacterium, it is what? Shigella bacterium, it releases that of the Shiga toxin itself, but in this case, the entero-invasive E. coli, it is never releasing any kind of the other toxin. Okay, it is not releasing such kind of any kind of toxin, means it is not releasing the Shiga toxin itself in that case. Okay, it is not releasing that of the Shiga toxin itself. So what is getting happened? It shows that of the fever, the abdominal pains, the watery diarrhea, these things they can get happened over here itself. Now the second one, we are representing it as of the enteropathogenic E. coli. We are representing it as that of the enteropathogenic E. coli or EPEC. Now these enteropathogenic E. coli, it will be showing that of the vomiting, diarrhea, along with the stools which are showing the, we can see that release of the blood also, that is a bloody diarrhea. This is showing over here, the which we are representing it as of the enteropathogenic kind of the E. coli. Okay, and the last one we are representing it as enterohemorrhagic kind of the E. coli. Now what is this enterohemorrhagic kind of E. coli? This, it shows the bloody diarrhea itself, stomach cramps it can get occurred, as well as watery diarrhea. So in all these things, the first two, these two, they are the most important one, and this enterotoxigenic E. coli, this will be the one which actually shows the release of that of two types of toxins which we are studying over here. The first one is the heat stable toxin. The second one is the heat labile toxins itself. Heat stable toxin means which can withstand high heat like 100 degrees Celsius for at least 15 minutes. Okay, and they are also acidic resistant one. And if I'm talking about the heat labile toxins, now these heat labile toxins are what? These are getting inactivated at that of 60. They are very sensitive towards that of the heat itself, and they can get inactivated at that of the 60 degrees Celsius after 30 minutes. Okay, at low pH. Now, this heat labile toxin, this is the kind of the bacterium which shows a close resemblance with that of the cholera toxins itself. Why? Because on the basis of the structure, it is showing that the resemblance. This one, it is also very much similar to that of the 5 subunits. It is consisting of 5 subunits, and it is responsible, all these subunits are responsible to bind to that of the toxin of that of the like epithelial cells itself. The toxin will be binding with that of the epithelial cells itself. Once it is binding to that of the epithelial cell, this will be showing the release of that of a specialized kind of the enzyme. That enzyme we are representing it as that of the adenylate cyclase enzyme. This adenylate cyclase enzyme will be, it will be doing what? It will be raising large amount of that of the cytoplasmic adenosine monophosphate, which causes that of the kind of that of these kind of abnormalities, whatever it is getting caused over here. Okay, that is the Escherichia kind of the toxin which is remaining present, and it is the 4 major categories as we had discussed. So students, you have to remember one thing, that this Escherichia is the kind of the bacterium which is actually the harmless one, which is not causing disease in the normal environmental condition. But once it is getting stimulated, it is responsible to cause, like I would say that, diarrheogenic kind of the symptoms itself, and which can cause many different kind of enteric infections also in that case. Okay, so these are like associated with that of the food, how they are coming within that of the food itself, by majorly by that of the fecal contamination of that of the water supplies and contaminated food itself, food handlers, okay, like that of okay, which are been frequently implicated, okay, in that of the EPEC, EIEC and ETEC itself. So EPEC, that means I would say that enteropathogenic kind of that of the E. coli, and then invasive kind of the E. coli, and that of the toxicogenic E. coli itself. In no, these are all the major outbreaks which it is showing, okay, and it is frequently implicated with him, and it is frequently been considered, or it is getting inside of that of the, I would say that the food itself in that case. So the food, it is getting contaminated by that of the fecal matter itself, and by that of the contaminated food handlers itself in that case. Okay, so this was about the Escherichia. The next group which we are going to discuss, a very important like genera, which we are representing it as the Clostridium genera. The species Clostridium is having a lots of species, among which this species, that is the Clostridium botulinum, this is specifically of mention because this kind of the species, it is responsible to cause a special kind of that of the like disorder or the infection, which we are representing it as the botulism itself. Okay, now from where we are getting derived this botulin word itself. Botulin words, it means what? It means sausage. Do remember the botulin words, it stands for that of what? Botulin words, it actually stands for that of the sausage itself in that case. Okay, so if I'm saying about the sausage in that case, that means what? It was first of all been observed that during the 17th century, I would say that it is showing the sausage infectivities itself. Okay, it was been found to be getting caused or remaining present within that of the sausage itself. Okay, so if we are saying about that of the sausage part itself, what is getting happened when the sausage, they are been obligately anaerobically they are getting prepared itself, automatically what is getting happened, this these bacterium, it is getting its favorable condition, and it shows its large amount of the infection itself within that of this thing. Okay, so Clostridium botulinum, the word we are getting it from that of the botulinum, botulic kind of the word itself, which stands for that of the sausage. Okay, so we'll see the features of the Clostridium. This is actually a bacillus kind of that of the like bacterium itself, the rod-shaped bacterium, and they are in the best manner, they are been isolated from that of the aquatic muddy kind of the condition itself. In the best manner, they are been isolated from that of the aquatic muddy kind of the condition itself, which are acting as a better source for the isolation of the Clostridium botulinum itself in that case. Okay, and it provides a moist as well as anaerobic nutrient rich environmental condition in which the Clostridium, it can easily flourish. It can easily flourish within that of this environmental condition itself. This is a gram positive kind of the bacterium. Okay, it's a gram positive kind of the bacterium itself. It's a motile bacterium which shows peritrichous flagella. Motile bacterium which shows that of the peritrichous flagellum itself. Peritrichous means the rod-shaped bacterium. It will be showing the presence of that of the flagella throughout that of its body structure itself. So it will be showing a peritrichous kind of the flagellum itself. And they are obligately anaerobes. These are actually obligately anaerobes itself. They are strictly anaerobe. Okay, they are not facultative anaerobe like there are the other microbes. They are strictly anaerobic kind of the microbes itself. So whenever any kind of the food which have been prepared in obligate anaerobic condition, or where the obligate anaerobic condition are being maintained, there is always an infectivity chance of that of this Clostridium within that of the microbe itself. Now we can say that these Clostridium, they produces specialized kind of the toxins, which we are representing it as the neurotoxins itself. Okay, and these neurotoxins, they are actually responsible for that of the botulism infection itself. Okay, so do remember Clostridium genera, other bacterium of the Clostridium, they can show the formation of the enterotoxin itself, but botulinum species, it is responsible to show the neurotoxin release itself. Okay, which directly attaches to that of a specialized nerve of that of our peripheral nervous system, and when it is attaching to that of our peripheral nervous system, it can even cause paralytic kind of the symptoms also over here. Okay, so it is growing at that of a minimum pH of that of the 4.7 and the maximum pH of 8.5 to 8.9. But now you have to remember one thing: in 4.7 or in that of this acidic pH itself, the neurotoxins are always in an activated state. But in the 8.5 and I would say that 8.9 condition itself, these toxin, they are getting inactivated over here. The toxins, they used to get inactivated over here itself in that case. Okay, students. So remember the toxin, it is inactivated in that of what? It is inactivated in that of, I would say, the part that is the alkaline pH itself, and it is activated in that of the acidic pH itself. As I said you earlier, this one is the soil saprophytic kind of the bacterium accordingly. Okay, that means what? It is mainly living in the mesophilic condition, in that of the soil condition itself. So we are representing it as the soil saprophytic kind of the bacterium. And it is growing in that of the alimentary tract of the animals as well as that of the birds itself. So now, from seeing about the difficult kind of the pathogenesis, the disease which it is causing, the kind of the food poisoning which it is causing, we are representing it as the botulism itself. Now, the toxin which is getting released with the same, that toxin, it is the neurotoxin, as I said you earlier, which is majorly affecting the cholinergic nerves of the peripheral nervous system. The cholinergic nerves of the peripheral nervous system itself. Okay, and this is having a very high mortality rate over here. It is bearing a very high mortality rate like that of 20 to 50%. Why? Because do remember, once this neurotoxin it gets inside of that of our body, it first of all binds with that of our nerve receptors. Once it is binding to that of our nerve receptors itself, automatically what will be getting happen? It will be causing that of the paralytic kind of the symptom. Once this is causing the paralytic kind of the symptom itself, automatically what will be getting happen? That will be causing the, I would say that the damaging kind of that of the effect itself within that of our body condition itself. So hence these are high mortality rate it is showing. Okay, and we can see that it is also showing initial symptoms of that of the part which occurs from that of the 8 hours to 8 days itself, and most commonly within 12 to 48 hours also the symptom it used to get existed. Okay, so do remember, once this botulinum toxin is inside of that of our body, what is getting happened? They're expelled from that of our intestine. First of all, they're going to that of our intestine. Once they're going to that of our intestine, from that of our intestine, they are getting released into that of our bloodstream. Once they are getting released into that of our bloodstream itself, through that of the bloodstream, these toxin, they reaches to that of our nerve. And once they are reaching to that of our nerves, it stops the acetylcholine flow itself. Once the acetylcholine flow from that of our neural axon region itself, it gets stopped, automatically information flow gets stopped. Okay, and ultimately individual can suffer from that of the paralytic kind of the conditions itself. Okay, so what can be the symptoms? The major symptoms are vomiting, constipation. Along with that, these are all the initial symptoms. Difficulty in swallowing along with dry mouth, dysphagia, and difficulty in speaking itself. That means dysphonia itself. These two things, they can get easily occurred over there. And this is the initial symptoms. Now, when these symptoms are primarily present and it is present for that of a longer time term period itself, the person can be invaded with the paralytic condition itself, and later onwards the person, they can die also. Okay, so this is a very lethal kind of that of the disease. It is having its lethal effect even at a very minor dose of that of 10 to the power minus 8 grams also. So this botulinum toxins are the most toxic kind of the substances itself, which is having their lethal dose within that of at a range of that of 10 power minus 8. So much lesser dose also it can cause a very detrimental effects within that of our body. They are high molecular weight proteins, these toxins, and these can be easily inactivated. As I said you, that these are generally found within that of what, these are generally found in that of like anaerobic kind of the environmental condition. I hope so you all had seen, while we are buying certain kind of canned tuna or canned fish or canned food products from that of the market itself, you all might have seen that within that of the canned products itself, okay, just beneath that of the lid, there is always an air space which has been maintained. Now, why this air space has been maintained? As because that air space ensures the presence of the oxygen. Once the oxygen is remaining present, it creates or it like causes that of the aerobic environmental condition, and under that of the aerobic environmental condition itself, this Clostridium will not be able to grow in a in a proper condition itself. Okay, it will not be getting a proper condition for that of its growth itself. So automatically what will be getting happened, automatically I would say that these bacterium and its growth will get stopped over here. Okay, so it is very essential that in the canned food industries, it is very essential to pack the food by maintaining proper, I would say that precautionary measures, so that an air space can be maintained. I would say that oxygenic environment can be maintained, so that these kind of the bacterium, it cannot grow within that of these kind of the canned food itself. Okay, so if I'm saying, we can say that association with the food. Now, how these bacterium, they are remaining associated within the food itself. There are 4 factors. First one, how can they get like ingested? Okay, there are 3 to 4 factors, I would say. The first factor, during the procurement. That means what? When the food products are getting prepared or are getting like harvested at that of the source itself, or they are getting, I would say that processed in that of the source itself. Okay, they can get what? They can, I would say that they can get infected by that of this bacterium itself. The second one, if the suitable conditions in food for that of for the suitable conditions in food for the growth of that of the Clostridium. That means what? Within that of the food itself, if anaerobic environmental conditions are being maintained, and all those anaerobic conditions itself, we can say that that will be causing the growth further of this Clostridium botulinum within that of the food itself. If this kind of the condition is there, the botulinum toxins, it will be getting like invested embedded within that of the food, and that can cause that of the the detrimental effects itself in that case. Okay, the second one, if the food is consumed, like we got the food from the market itself, we're opening the food and we are starting it to consume without proper heating or warming the food itself. When the food is consumed cold or after a mild heat treatment itself, that kind of the heat treatment, it is not sufficient enough to inactivate the toxin. Automatically the botulinum toxin will be found within that of that particular group itself, and hence it can cause a disease. So you have to remember that the botulinum toxin, it is what actually I would say that it is found typically in that of the anaerobic kind of the environmental condition, like the obligate anaerobic condition, and we have to ensure the presence of that of the aerobic conditions itself, so that these organisms, they can grow properly. Okay, these organisms, they cannot grow properly itself, and they cannot multiply within the reservoir. Okay, so for that reason, the canning food industry or the canning food preservation industries are required to maintain an extra precautionary care while processing the food or while packing the food itself. Okay, students. So that's all for today. We had discussed about that of the Escherichia and that of the Clostridium. Okay, we are going to bring some of the other microbes in the later day tomorrow itself, and we are going to discuss some of the other topics also in the coming onwards classes itself. In the meanwhile, if you need notes and other things, you can contact over this number itself. And if you're having any doubt, you can put in the chat over here in the discussion itself. We are always there with you. IIOCS is always there with you to pick you, to keep you to that of the your confidence boosted up and to take you to that of the pick itself in that. Okay, students. Thank you, everyone. Thank you. Stay well. Okay, study well and keep your confidence level always high.